Okay, so this is backed up a little bit from what we got to on in last class on Tuesday, but I wanted to review this again and then we'll go forward and finish up photosynthesis. So photosystems within the chloroplast of a plant, embedded in those thylakoids, so those little sacs, the green sacs there, are things called photosystems. Photosystems are what's actually going to absorb the light energy. And it's not the photosystem as a whole, it's actually the pigments that are embedded in the photosystem. So pigments such as chlorophyll, so there's chlorophyll written there on the top, so the central chlorophyll molecules. Pigments such as chlorophyll will absorb light energy. And remember, pigments are molecules that absorb some energy, they reflect some other energy. You are going to see the wavelengths of light that are reflected. So a green object is going to look green because it's reflecting green and it's absorbing purple and blue and red and all the other colors. So light energy hits pigment molecules in a photosystem. That light energy is absorbed by the pigment and then it is passed off to a neighboring pigment molecule. And it all gets funneled down to the central pigment molecule. So in your photosystem, you're going to have all these green pigment molecules. So that's a dry marker. It's not very useful. So you're going to have all these chlorophyll pigment molecules, and as they get hit with light, here we go. They get hit with light, they're going to transfer that light energy to neighboring pigment molecules. So light energy hits this one, it's going to funnel its way down. Light energy hits this one, it might go this way. Light energy hits this one, it might go this way. So eventually all the light energy that hits these outer pigment molecules is funneled down to this central chlorophyll molecule. So this is your central chlorophyll A molecule there. And when that happens, this chlorophyll molecule sends an electron up to an electron receptor up here. So an electron is knocked off that central chlorophyll molecule, sent up to the primary electron acceptor. Okay? So that's how light energy will energize an electron. So you've got an electron up here that is energized. Okay. So I went over this last class, but we're going to go into it in a little more detail and break it down into several steps. So that happens twice in most plants. Some plants only have one of these two photosystems, but a lot of them have two, and we're going to talk about it as two and kind of leave it there. So this first photosystem, which is called the water-splitting photosystem, this process happens. Energy is funneled down to central chlorophyll molecule, energizes an electron. That electron is sent up to the primary electron acceptor. This central chlorophyll molecule now is missing an electron, so it's positively charged. And it's got to get another electron from somewhere, or else this can't fire again. So it's got to reload. It's got to reload with an electron so that it can happen more than once. You don't want to just use the photosystem once and be done. The plant wants to continually use that photosystem. So what happens is water is split and sends an electron to that central chlorophyll molecule. Okay. So here's that first step. Photons excite electrons in the first photosystem, transferring the electrons to the primary electron acceptor, and then the electrons are replaced by splitting water. So that's step one. So now you have a highly energized electron, or pair of electrons, in the primary electron acceptor of the water-splitting photosystem. So we have energy we can use to do something. And the first thing that plants are going to do with the energy from the water-splitting photosystem is they're going to generate ATP. And do you remember from Tuesday how ATP is generated in this process? Okay. Yep, so it's going to use ATP synthase by first setting up a hydrogen ion gradient. Just like we saw in the mitochondria, same thing is going to happen in the chloroplast. And that involves an electron transport chain. So that highly energized electron that's in the primary electron acceptor is going to enter an electron transport chain. The electron transport chain is going to generate ATP. And the way it's going to generate ATP is the same way we saw in cellular respiration. You have pumps that are going to take hydrogen and they're going to pump it across a membrane to create a ion concentration gradient. And so this is your membrane. It's going to pump hydrogen across a membrane. In this case, it's going to be pumping it into the thylakoid, but we're not going to be concerned too much about that. So really, if you just remember that the electron transport chain is going to generate ATP by pumping hydrogen across a membrane, that's really good for now. Okay, so ATP is produced that way. That's one of our products of the light reactions of photosynthesis. So the second thing that's produced by the light reactions of photosynthesis is NADPH. And that's produced by the second photosystem, basically. You have your NADPH producing photosystem. Light, again, will hit the reaction center, or hit the photosystem, funneling energy down to the reaction center chlorophyll of the second photosystem. That will then excite electrons, sending them to a primary electron acceptor, and those electrons will combine with NADP to create NADPH. And again, just like your first photosystem, this photosystem is going to need to replace its electron from somewhere. It's now lost an electron. So it's going to take it from the electron transport chain. So the electrons coming off the electron transport chain after they've been used to produce ATP will enter that second photosystem and be used to create or to replace the lost electrons in the NADPH producing photosystem. Okay. So, any questions on this process? Do you have a good understanding of how ATP is produced and how NADPH is produced? Those are your two products of the light reactions. Could you explain how energy is captured by a plant? How a plant captures energy. So how does a chloroplast capture light energy? So if I asked you that question, would you be able to answer it? Anybody? Any guesses? What's the first step?
Light.
Okay, what's light going to hit? What does light energy hit? Okay, what part of the leaf? Okay, it hits the chloroplast. What part of the chloroplast? Okay, which? The reaction center? Yeah, so it's going to hit this, which is, the whole thing's called a photosystem. So it's going to hit a photosystem. How does a photosystem capture light energy? How does it convert energy from the sun into something that can make ATP and NADPH?
Absorbs and reflects it in its pigments.
Okay, so it's pigments. So it's pigments within the photosystem will absorb some of that light energy. And then what happens to that energy?
After it's absorbed, it's funneled down through.
Okay, it's funneled down to the central chlorophyll molecules. And what happens when all that energy hits the central chlorophyll molecules? Okay. Electrons, what did you say? Sorry. Oh, that's, you're getting ahead of us a little bit. So what happened, why do the electrons, so the electrons have to come off water to replace electrons. What happens to the electrons in the central chlorophyll A molecule? They're sent to the primary electron acceptor. Okay, so now central chlorophyll A molecule has lost electrons, and then what happens? So water is split, and the electrons are replaced by water. So you need the electrons to be missing first before the electrons can be replaced. Right? So light hits pigment molecules. That energy is funneled down to a central pigment molecule. All that energy is so strong that it causes an electron to be transferred, and then that electron is replaced by the electron off of water. So now you have a high energy electron in the primary electron acceptor. What's that going to be used to do? Okay, we'll power the electron transport chain, which ultimately produces what? ATP. Okay. These are really all the concepts I want you to get out of this. So just try really to get this. Okay, so that's what this water splitting photosystem does. It excites electrons by the photosystem funneling energy, goes to the primary electron acceptor, which powers the electrons, then power the electron transport chain, which makes ATP. What happens in the second photosystem?
Light reactions generate ATP and NADPH.
That's the overall thing, right? But what's produced by the second photosystem? So the first one, we said, produced ATP by energizing electrons that run through the electron transport chain. What happens in the second photosystem? This one on the right. All right, produces NADPH. Right? It's all up there. So again, just like the first photosystem, energy hits the photosystem pigments. That energy is funneled down to a central pigment molecule. All that energy is high enough that it pops off an electron. The electron goes up and gets added to NADP to make NADPH. Where does the NADPH producing photosystem replace its electron from? Like, where does it get another electron to replace that lost electron?
Splitting water again?
Nope, water doesn't get split again. So how do electrons get back into the NADPH producing photosystem? That was very mumbly. What did you say? No, go ahead. No?
Light?
You don't. No, the electrons don't come from light. Where might we have an electron that's already used up all its energy?
Chloroplast or the chlorophyll?
Well, it originally came from chlorophyll, yeah.
Transport chain.
Yeah, the electron transport chain. So your electron transport chain has electrons that have used up all their energy generating ATP. They will now replace the electrons in the NADPH producing photosystem. Okay, so feels like you really need to review this a lot. So this is not going into excessive amount of detail, but it's just kind of this sequence of events is really important for photosynthesis in order to produce the two energetic products of the light reactions. So what are the two products of the light reactions?
ATP and NADP.
NADPH, yeah, all right. So all you guys had to do was look at the board. Are you paying attention? Or are we all falling asleep? Is it that bad? Right. Okay. So what's going on? Do you actually understand this or not? not at all. No? I don't know. All right. It's not that bad. There's only like 4 or 5 steps that I'm trying to get. Yeah? All right. So we'll keep going over it. You'll eventually get it. Okay. All right. We'll get there. All right. All right. So I will show you a video in a little bit, and that'll summarize it again. But really, review this, so that if I ask you questions about it, you should be able to answer it. So if I say, how is ATP produced by the light reactions, you'll say, energy is captured by a photosystem to excite an electron. That electron powers electron transport chain, which generates ATP. That should be something that comes quickly to you. And then if I say, how is NADPH produced? You say, well, energy is captured by a photosystem to energize electrons. Electrons are then transferred to NADP to make NADPH. That's the whole thing. That's really the basics of this whole process. They say, where do electrons originate in the first that replace the electrons lost by the first photosystem? They say splitting of water. They say, where do the electrons come from that replace the electrons lost by the second photosystem? They say electron transport chain, something like that. So, okay, those are kind of the concepts I want you to get. Okay. So now we've already talked about this a little bit. So hopefully this is a review to some extent. This is adding in a little more detail on that electron transport chain. So here, this whole gray thing here is one thylakoid. And a thylakoid is a little sac, basically. So if this was a thylakoid membrane, this thylakoid, would be something like this. Right? It'd be a big sac here. And between the two photosystems, this is your electron transport chain. Your electron is passing through the electron transport chain, and as it does, hydrogen is being pumped into the stroma, or the inside of the thylakoid, so from the stroma to the inside of the thylakoid. So here is all your hydrogen. So there's a lot of hydrogen here, not very much out here because it's pumping it all in. So you have ATP synthase. And what's going to happen to this hydrogen that's built up concentration inside the thylakoid? Yeah, it's going to flow through ATP synthase to power it, right? And this is going to convert ADP into ATP, which is what we wanted to produce, right? So that's what this graphic shows. So you've got ATP synthase, which is the purple blob on the right there. You've got your two photosystems, and between them, the electron transport chain. The energy from that first photosystem is powering the electron transport chain to pump hydrogen into the thylakoid, and then as they diffuse back, they're going to diffuse back through ATP synthase. So ATP is produced here, the same mechanism that produced ATP in cellular respiration by diffusion through ATP synthase, diffusion of hydrogen through ATP synthase. Okay, so that's the light reaction. We've produced 2 byproducts. We've produced ATP, we've produced NADPH. Both of those are going to go to the Calvin cycle to create sugar. Okay, so before I talk about that, let me pass this around. But questions on this? Are you feeling a little more like you understand what's going on in the light reactions, just at least a bit? If not, then you need to be asking me questions so that we can clarify it more. So the first photosystem produces which? ATP or NADPH? Yeah, it produces ATP. So it's what generates the power for the electron transport chain to generate ATP through the concentration gradient. Second photosystem, what does that produce? NADPH, right? Here we go. Good. All right. A few of you are getting there. Okay. Before we go on to the next step, which is using this energy to make sugars, why do you think plants make sugars? So we just produced some ATP, and really ATP is what powers cells to do stuff. So why don't plants just live off of this ATP? Why do you think they use this ATP to make sugars? Okay, so they're benevolent and they want us to be able to eat potatoes and things like that. Maybe some of them think that way. I don't know. I doubt it. Really, unfortunately, life doesn't work that nicely. They don't exist for us to eat. What do you think they want to do with those sugars, though? You're right. Something wants to eat it. What?
Produce something for themselves?
Yeah. Really, it's more for themselves. So yes, they're producing it to be eaten, but it's to be eaten by themselves. So think of trees up north that lose their leaves in the winter. Are they able to generate energy from sunlight during the winter? No, they've lost their leaves, but they still need to survive. So they're going to survive off of stored resources. So sugars that they've produced during the rest of the year, they'll be able to use those. Right? Think about plants at nighttime. They can't work off of sunlight all the time. They need to be able to have something to generate energy at night when the sun's not out. So kind of an analogy to this is if you've ever used, let's say you use a solar-powered calculator and you put your thumb on the solar panel, the calculator turns off, right? That's something that runs directly off of the light, right? And it can only be used when there's light out. Thankfully, you're probably not going to use that calculator in the dark, because you wouldn't be able to see the numbers anyway. So something like that, it's fine that it works just straight off the light. But there are other things that you don't want. So let's say you have solar panels on your house and you want to generate electricity for your house. You probably are going to most likely use that energy more at nighttime. Like you might use it to turn on lights at night, or you might use it to be able to charge batteries and things at night. So what you'll do is during the day, when there's lots of sun out, you have those solar panels on your roof, you're charging batteries, and then your house runs off the batteries at night. It's the same thing for a plant. During the day, when there's lots of sun, they're capturing sunlight energy and they're producing sugars. And then when they can't, when they need that energy, they're going to convert the sugars back into energy and use it. So they're not running directly off the sun, they're running off of a storage product of the sun energy, which is sugars. For us, as consumers, you're right, we do benefit from this. We steal the sugars that the plants were going to use for themselves. So, maple syrup is a prime example of this. There's something in the spring called, the sugar rush, where the trees actually start mobilizing a lot of the sugar. So they actually do go pretty dormant during the winter. And then right when they're about to start growing again in the spring, the sugars start flowing through the tree to get to the locations, to get to the branches so they can start building leaves, things like that for the spring. So we tap a spigot into the tree and we siphon those off and we make maple syrup out of them. We do the same thing with a lot of the foods we eat. So carrots are a storage location for lots of sugars. That's why they taste really good. Potatoes are a storage location for a lot of sugars. So all the things that we like to eat, fruits, vegetables, carrots, roots, and tubers like potatoes, those are all locations where plants store their fuel. And we take them and we eat them. So it's... So we benefit a lot from photosynthesis as well. Okay, so they've made these energy products, NADPH and ATP, but they want to convert them into a storage product. They want to convert it into sugar. And the way they do that is through a process called the Calvin cycle. And really, that's almost enough for you to learn, is that the Calvin cycle converts carbon dioxide into sugar. It's kind of like the Krebs cycle, like the citric acid cycle that we talked about in the last chapter, but in reverse, kind of a reverse form of the citric acid cycle. Citric acid cycle, you took in a sugar form, you took in acetyl-CoA, and your output was carbon dioxide and ATP and NADH. Right? Here, you're going to take in carbon dioxide, you're also going to take in ATP and NADPH, and you're going to generate a 3-carbon sugar that will eventually get converted into a 6-carbon sugar like glucose. And it's a cycle, which means that the sugar that it starts with in there gets converted and then gets replaced in the end. But really, all I really want you to know about the Calvin cycle, because we've already taken in enough information, I think, is that the Calvin cycle will convert the carbon dioxide in the air into sugar using the energy from the light reaction. So using ATP and NADPH, the light reaction energy, it makes sugars from carbon dioxide. Okay? All right, I'm going to show you a little video. This is a video from the 1010 version of your textbook. So it again goes into a little extra detail that I'm not going to hold you to. I've already taught you kind of more detail than I'm probably going to expect you to recall. But you'll get the idea of the whole process, and it kind of puts it all together nicely with nice pictures. So watch this again, and hopefully this all sounds like review to you at this point. Okay.
Plants provide us with food to eat and oxygen to breathe. They perform this amazing feat by the process of photosynthesis. Let's take a closer look. Photosynthesis requires carbon dioxide, which diffuses into the leaf through small pores and then enters the cells. Inside the cell, carbon dioxide diffuses into the chloroplasts, where photosynthesis takes place. Chloroplasts use energy from light to transform carbon dioxide and water into sugar and oxygen. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. Here, light energy is converted to chemical energy in the first phase of photosynthesis, the light reactions. The 2 green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the 2 photosystems. Notice the small mobile electron carriers that shuttle electrons from one large complex to another. Now let's take a closer look at the steps of the light reactions. The photosystem on the left absorbs light energy, exciting electrons that enter the electron transport chain. Electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, into the thylakoid. In the photosystem on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of hydrogen ions inside the thylakoid powers ATP synthase, producing ATPs. The light reactions in the thylakoid have produced 2 energy products, ATP and NADPH, that will now power the production of sugar in the Calvin cycle. The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With 3 simple ingredients, carbon dioxide, water, light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism and ultimately providing your fuel as well.
All right, so that's really an overview of all of photosynthesis. So you have the light reactions, which took light energy in, captured by photosystems to excite electrons. Those electrons were then used to, 1, generate ATP by powering electron transport chain, and 2, generate NADPH, which is an electron carrier. Then ATP and NADPH went to power the Calvin cycle, which converts carbon dioxide into sugar. And that's really the big overview of all of photosynthesis. Is one part generates energy, ATP and NADPH. The other part uses that energy to make sugars from carbon dioxide. Right? Okay. So I just have this is kind of an overall summary of why photosynthesis is important. And then we're going to talk a little bit about plants that live in extreme climates and some differences that they have. And they're just slight differences from what we just talked about. And that'll pretty much be it for the photosynthesis chapter. So photosynthesis takes energy as sunlight and stores it as a chemical energy in organic compound. So it's a hugely important process because the sugars made by the chloroplasts are now able to supply chemical energy and carbon skeletons for most organic life. So they're going to make the basis for things like cellulose, which makes the structures, the overall structure of a plant. So that's what makes cell walls in plants and gives them their structures. It can also be converted into things like chitin, which are important for animals' structure. So exoskeleton, a lot of bugs, is made out of carbohydrates. So any carbohydrates, this is how they originate. So we're making organic molecules and energetic molecules. So plants will then store their excess sugar, so anything that they don't use to either build on themselves or use immediately as energy, they will store as starch primarily. And starch gets stored in structures such as roots, like carrot, tubers, which are like potatoes, so below ground storage products, seeds and fruits. So all the things that you like to eat, all the plant parts that you like to eat, for the most part, you like to eat them because they have starch in them. They have a storage form of a sugar. Fruits have other forms of sugars as well. They have fructose, like that. So that's mainly why we like to eat those things. That's where we get our energy from. And then in addition to food production, photosynthesis also produces oxygen as a byproduct. So when water is split, when this water is split to replace the electron lost by the central chlorophyll molecule, this gets split into H plus oxygen, so O2 here. So we get oxygen gas as a byproduct. And then oxygen is then released by the plant, and we can use it. And we need it in order to allow us to break down the sugars that the plants produced and convert those into ATP, which our cells can then use to power muscle contraction or power active transport or any of the other tasks, making proteins and transporting proteins, things like that. So photosynthesis, hugely important, really important thing to happen. Amazing that plants can do this with only sunlight and water and carbon dioxide. That's really the main things they need. So other things that you give to plants to help them grow helps them build certain parts of their bodies, but the main components that are needed to make sugars is just carbon dioxide, water, and sunlight. Right. Okay. So this next slide, I think this next slide is the information that's in this next slide I've taken from another course. So it's a little bit, again, more detailed than I want you to focus on, but it'll help you get the idea of this next topic. So I only gave you kind of a summary slide, which is the next one that I'll show. What we just talked about, the light reactions followed by the Calvin cycle in the manner that we spoke about it, is known as a C3 pathway. And don't worry about why C3, and actually don't even remember C3. That's not hugely important for this. But it's called C3. There are other kinds of ways that carbon enters, and they have names such as the C4 and the CAM pathway. So C3 is carbon dioxide enters, is incorporated through the Calvin cycle, and that's what we were just talking about. These other pathways, C4 and CAM, exist because when plants are stressed by environmental conditions, when they live in hot, dry climates, so desert-like climates, anywhere where it's hot and dry, a really weird thing happens to plants. If you look on the right here, this is from several slides ago. I showed you this image. It's from your textbook. It shows the openings on the bottom of plants, and I pointed this out the other day. So the stomata on the bottom of the plant is where carbon dioxide enters and where oxygen leaves. And you saw that in that video. It's the opening where gas is exchanged. The stomata are also the only location on a plant where water can evaporate. So the rest of the plant is covered in a waxy coating that prevents water from leaving, water from evaporating. So stomata, those holes, are a source of water loss. So if it's really, really hot and really dry, in order to protect itself, to keep the plant from drying out, from dehydrating, plants will close the stomata. So that prevents water from leaving, but it also keeps them from getting carbon dioxide in and releasing oxygen. And this is something I don't want you to really focus on. It's not something I'll test you on, but it's a kind of a crazy concept. There's a process called photorespiration, whereby plants that have a high level of oxygen in their cells and a very low level of carbon dioxide, rather than pulling carbon dioxide into the Calvin cycle, they start pulling oxygen. And we talk a lot about this in 1010. We talk about this enzyme called Rubisco, but don't worry too much about it for this. I don't want you to get bogged down in the details there. But basically, just hot and dry climates for most plants, very bad. It causes breakdowns in the photosynthesis process and makes it so they can't produce sugar. That's really the end result. So certain plants that live in environments where they're going to get hot and dry days a lot have to come up with other ways of generating sugars besides this C3 pathway, because when they close their stomata, they wouldn't be able to produce sugars. So for plants around here, you don't get very many hot, dry days. So when it's hot, it's usually pretty wet, right? And during the winter, when it's drier, it's usually not all that hot, right? So hot, dry days are pretty rare in an environment like we have around here, but in an environment like the desert, it's going to be a lot worse for them. So here are these two alternate pathways, and I'm just going to briefly go into what happens. CAM and C4 plants, what they'll do is the take-home message is they are alternate ways of feeding carbon dioxide or feeding carbon to the Calvin cycle to let them survive better on hot, dry days. So what C4 plants do is that they pull carbon dioxide into the plant in a cell where the Calvin cycle doesn't doesn't happen. So they have one cell type where they will actually pull carbon dioxide in, and they have an alternate way of converting it into a 4-carbon sugar. And then they feed that 4-carbon sugar to the cell where the Calvin cycle is happening. And so the light reactions don't occur in cell 2, and there's no oxygen buildup in cell 2. There's no depletion of carbon dioxide because they constantly feed that cell with CO2 coming from this 4-carbon compound. So C4 plant, like sugarcane, takes carbon in a separate cell than it does the Calvin cycle. Cam plants, like on the right, like pineapple, they separate their Calvin cycle from what's called carbon fixation, from bringing carbon in, by time of day. So they will open up their stomata at night, they'll pull in carbon dioxide, and they'll convert that carbon dioxide into a 4-carbon sugar all night long. At nighttime, it's generally not as hot, there's not as much sunlight, so there's not going to be a lot of evaporation, so they're not going to lose as much water at night. And then, after they've built up this huge store of this 4-carbon sugar, they'll close their stomata down during the day, and they'll feed the Calvin cycle that 4-carbon sugar during the day. So both of them have ways of separating the location where you're going to get high oxygen, low carbon situation from the Calvin cycle, which that would be detrimental to. So you have separating it by having 2 different processes in 2 different cells, and you have separating by having something happen at night and something during the day. Okay, so I don't want you to focus on the specifics of these. Just know that the CAM and C4 plants are alternate ways of dealing with getting carbon to the Calvin cycle when it's hot and dry out. So you might wonder, well, looking at these, well, why don't all plants do it this way? Why do some of them have this mechanism that we first talked about that breaks down completely on hot and dry days? Well, it's because there's a trade-off. The C4 and the CAM pathways require extra energy to do this. So in order to make that 4-carbon sugar, either in the separate cell in the C4 plant, or to make that 4-carbon sugar at night in the CAM plant, they need to burn ATP to drive those pathways. So they're less efficient than a C3 plant, yet they can survive in hot, dry climates. So this happens a lot, and we'll talk about this more when we get to kind of the ecology section of the course. But a lot of organisms have trade-offs in their lives. They have to decide, do I do this really well, or do I do it slightly less efficiently, but can I do it in more environments? So the CAM and the C4 plants can survive in a broader range of environments. They can survive in the desert, they can survive with ample rain, but they grow a little slower. So this is why if you grew a cactus in your house, it's not going to grow very fast, right? But you can forget to water it for a few weeks and it'll survive, right? But if you were to grow some kind of vine next to it, the vine would quickly outpace the cactus in terms of growth as long as you were watering it. But as soon as you stop watering that vine, it's probably going to die, right? So neglectful people, you want a CAM or a C4 plant as your house plant because they're hard to kill. They survive that stress of not being watered for weeks on end. But they're going to grow very slowly. You're not going to see much growth in a cactus over the course of a year, right? Okay? Okay, so that's the end of the photosynthesis chapter. So things to focus on that general idea of photosynthesis. We focused a lot on the light reactions, so that's something I expect you to understand a little bit better than the rest of it. For the things like the Calvin cycle, I just briefly talked about it. Really just know the input and output for that. So carbon dioxide in, energy in, ATP in, NADPH, and sugars come out. And that's really that. And then briefly, the only thing about the CAM and C4 plants is that they can survive in hot, dry climates, but it requires a little bit more ATP investment. So they're a little bit less efficient. Right? So really, you can boil the whole chapter down to just those few things. Any questions on photosynthesis on this chapter? So definitely spend some time reading through your notes, watching that video again when you get a chance would be a good way to go about it. Okay. So that was chapter 7. For the last little bit of class, I just want to start on chapter 8 so that we get a little bit ahead from where I said we'd be on our schedule. That'll give us a little bit more breathing room when we get to chapter 9 and we start talking about genetics, and I'll be able to spend a good amount of time just doing some worksheets in class and doing some practice genetic problems that I think most people find pretty interesting, and we'll probably do more of those on the next exam than the rest here. So chapter 8 is all about cellular reproduction. We're going to talk about 2 processes, and we won't get to the second one today, but we'll touch on the first one. We're going to talk about a process called mitosis and then a process called meiosis. So cells divide. We know this. You started your life as a single cell. So your father's sperm and your mother's egg combined, they created a zygote. That's a single-celled human. You are no longer a single-celled human. You now have millions of cells making up your body. So somehow that one single cell had to replicate itself, form 2 cells. Those 2 cells had to become 4, 8, 8, 16, and so on and so forth until you had all the cells that make up your body. That's a really cool process to study. If you do go on and take more biology classes, if you eventually take a developmental biology class, you'll learn about how that one single cell has grown and then differentiated, become skin cells and heart cells and eye cells and lung cells, and how they all take on those different roles. Very cool process. We're not going to talk about that right now, but we are going to talk about just general cell division. How does one cell become 2? So cell division is required for growth. That's kind of what I was just talking about, so that one on the right there. So going from an early human embryo up to a full-grown adult. But it's also responsible for cell replacement. So when you injure yourself, so if you get a cut, your skin cells need to divide and replicate so they can patch up that cut. And you probably know this by now, but different cells will divide at different rates. So if you get an injury in your cheek, the inside of your cheek, that's going to replace pretty fast. Your gut cells and all the cells basically for your whole digestive tract, they pretty much replace themselves daily. So every day you get a new lining almost. And that's because as you eat, your food scrapes along the insides of your digestive tract and it breaks off a lot of those cells. That's why CSI shows, they show them taking a swab from the cheek. Cells on the cheek fall off when you touch them, pretty much. They're really easy. They come right off. This is why in your saliva you have lots of cheek cells, and they're easy to catch, right? But things like nerve cells, I think, as an adult, the estimation is about once every 100 years that they'll divide. So pretty much never. So once you are an adult, if you damage a nerve cell, it's not coming back. It's not going to repair itself. So you can damage your cheek cell and it's repaired the next day. You damage a nerve cell and you no longer have the function of that nerve cell. So that's why you have paralysis. It's a pretty common thing that can happen. If you break your spinal cord, you can no longer walk. So there's a lot of research being done to try to figure out, well, why is this? Why do some cells divide really rapidly? Why do some not divide really at all? And can we reprogram cells to divide more rapidly? So could we give you a drug that would cause your nerve cells to divide again? And then we could repair those kinds of problems. So that's something that researchers are working on. So there's a lot of research going on in mitosis to try to solve those kind of problems. Can we grow cells of a certain type? Can we cause things to be grown that just in a petri dish or something like that? So there's a lot of kind of aspects of this. So cells divide. So the first part of this chapter is all about how this happens. And one of the main things that is controlled during cell division is DNA. So DNA, which you know is found in the nucleus of your cells, is the thing that's kind of most important to get right in cell division. Is you want to make sure that when you take one cell and you make two cells from it, that each of those cells that you make has the same set of DNA. Because DNA is an instruction manual for how to make proteins, and in order for this cell to function as well as this cell, they both have to have the same set of instructions. So DNA is going to be divided, it's going to be duplicated, and then it's going to be divided during cell division. And mostly what we're going to talk about in meiosis is division of the DNA, ensuring that the same amount of DNA goes to both cells. So one thing that makes it easier for DNA to move during cell division is the way that it's packaged. So just like if you were going to move out of your apartment into a new apartment, you would take your belongings and you would pack it up, put it in a box, and then pack it in a truck and move it. You wouldn't just take all your belongings and shovel them into your car, right? Because you'd probably lose some stuff and you'd break some stuff. DNA does the same thing. DNA is wrapped around proteins and tightly coiled prior to division. So eventually, probably next class, I'll show you a video of cell division. You'll see that the first thing that happens is the DNA coils up and becomes really tightly packed. And those packages of DNA are called chromosomes. So we don't just have one long strand of DNA in our cells. We have packets of DNA. And humans have 46 chromosomes. You can see on this chart, this is a chart of the number of chromosomes for different organisms. And you can see the number of chromosomes has nothing to do with intelligence or size or anything like that. Giraffes have 30 chromosomes. Koalas have 16. Rats, there's a certain type of rat that has 102 chromosomes. So it's just the number of packages our DNA is broken up into. Our DNA is broken up into 46 packages, so 46 volumes. Right? So during cell division, each one of these 46 chromosomes is going to first be replicated or duplicated, and then it'll be separated. So we're going to see, we're going to talk about something called the cell cycle, which is really the whole cell's life cycle from when it first is created to when it divides again. During one part of the cell cycle, we're going to see duplication or DNA synthesis. And what happens is your chromosomes will be duplicated. At that point, each copy of the chromosome is called a sister chromatid. And then through the process of mitosis, which we'll talk about, those sister chromatids are going to be separated, and each one will be put in a new cell. Those new cells are going to be called daughter cells. So replicated chromosomes have two sister chromatids. A replicated chromosome is a chromosome that hasn't been divided yet. It's been replicated but not divided, and it has sister chromatids. And then each chromatid is going to become a new chromosome in each of the new cells. So we'll go from having 46 chromosomes to 46 replicated chromosomes to 2 cells, each with 46 chromosomes by the end of mitosis. So the goal of cell division is to make identical cells. Okay. So when does this happen in a cell's life? What happens in something called S phase of the cell cycle. So broadly, the cell's life can be separated into 2 components. One is active cell division, which is called mitosis, or the mitotic phase, and one is called interphase, which is the rest of the cell's life. So all the stuff we've been talking about, cellular respiration, photosynthesis, protein synthesis, where it's going to be transporting protein in those vesicles, that all occurs in interphase. Interphase is about 90% of a cell's life, most cells. It can be even more. So your nerve cells, it's pretty much 100% of their life. The mitotic phase, which is about 10% of the time, is that very short time period that a cell is actively splitting into. So when a cell is not actively dividing, it's in interphase. And interphase, it's either growing, which is the G part of interphase. There's G1 and G2 on there. Or it's replicating its chromosomes, and chromosome replication occurs during S phase. Okay, so we have G phase, and we have S phase of mitosis. Okay. So during S phase, chromosome will duplicate. You'll have two chromatids. And then where in the cell's life do you think the chromatids will be separated? Which part of the cell cycle do you think is where the chromatids get separated and you then have two separate chromosomes in new cells? Nope, G, so you're narrowing in on it. So interphase is growth and duplication of the chromosomes. So what's that other phase there? So the S phase is where is synthesis. That's where DNA is going to be replicated. But it's the bottom phase there, the mitotic phase, that's actual cell division. So yeah, that's good. Thank you for answering, though. So you have interphase is responsible for normal cell metabolism, but the S part of interphase is where you're going to be taking your single chromosomes and replicating them, so they have chromatids. These chromatids aren't going to divide until the mitotic phase, that bottom phase, the darker colored region. So the mitotic phase is actual cell division. Mitosis is going to separate your chromatids and make two new cells. Okay. So basically, the most important part of mitosis, or most important part of cell division, is separation of chromosomes. And so we're going to focus on the mitotic phase and focus on this separation of those sister chromatids. And I'm actually going to break it down into several distinct little phases within it. However, the majority of a cell's life is not spent dividing. The majority of a cell's life is spent doing cell stuff, doing respiration, doing photosynthesis, making proteins, transporting proteins, things like that. And that all happens in interphase. So that's the real take-home here, is that interphase is where a cell is being a cell. Mitosis is when a cell is actively dividing, and mitosis is a very small portion of its life. So from this chapter, the big thing I want you to get, at least from mitosis, is that if I show you pictures of cells, you'll be able to tell me what stage of mitosis they're in. So we're going to go through the different stages of mitosis, and then if we have time, which we might not today, but we'll do that definitely next class, I'll show you some pictures of cells in different stages, and you should be able to identify which one they're in. So there are four stages of mitosis that I want you to know. And there's a mnemonic for remembering these. So interphase is kind of outside of mitosis, so that's separate. That's why it's a different color. And then there are four stages of mitosis. There's prophase, metaphase, anaphase, and telophase. And one way to remember this is the mnemonic, pour me another tequila. It's kind of a fun one. There are lots of other ones there, but that one's always stuck with me since college. So you're welcome to use that one, pour me another tequila. That'll help you remember prophase, metaphase, anaphase, and telophase. And so I'm just going to briefly go through what's happening in the different stages, but really what I want you to be able to do is, if I show you a picture, be able to identify that stage. So interphase here, where the cell is doing cell stuff, your chromosomes are all unraveled. And the reason for that is that DNA is instructions for how to make proteins, and you can't read those instructions if it's all tightly bound up. So it's like you can't read your recipe book if you don't open the pages of the book. So during interphase, the DNA is going to be all unraveled, and that's why you just see this big purple blob there with the DNA unraveled. During prophase, DNA is going to condense. And so you're going to go from early prophase, which is this graphic on the bottom where you still see just a big blue dot, to kind of a later prophase on the right there where you start to see individual chromosomes. So they're really kind of clarifying, and you can see little spindly lines there. So what's happening is the chromosomes are condensing. They're getting coiled up, getting packaged up during prophase. Another thing that happens is that the machinery that's going to help those chromosomes eventually divide is growing. And that's called the mitotic spindle. And it involves centrosomes and some cytoskeletal elements called microtubules, which we briefly talked about a few chapters ago. So those microtubules will eventually attach to each of the chromosomes and pull them to where they need to be to divide. So part of prophase is this machinery starting to be produced. So mitotic spindle is forming. So chromosomes are condensing, mitotic spindle is forming, that's prophase. Eventually, those microtubules are going to attach to all of the chromosomes. So in humans, that's 46 chromosomes are going to be attached to microtubules, and they are going to pull those chromosomes towards the center of the cell. So when all the chromosomes are found in the center of the cell, this is when we've entered the second phase, metaphase. So metaphase, the chromosomes will line up along the center of the cell. And you can see in the bottom picture that in a real cell, an actual cell, it's not quite as neat, but they're still all lined up near the center. So the blue there are the chromosomes. The green is the mitotic spindle fibers. Those are the microtubules. Right? So metaphase, chromosomes line up on the center of the cell. As soon as that happens, those two sister chromatids will start migrating towards opposite ends of the cell. So up until now, you've had your replicated chromosomes with two sister chromatids. After metaphase, when it starts entering the next phase, anaphase, those sister chromatids will separate and head towards opposite ends of the cell. So that's what anaphase is. And you can see you're getting these two clusters of chromatids moving towards opposite poles. Once those chromatids, which are now considered daughter chromosomes, reach the opposite ends of the cell, they'll start to unravel. A nucleus will begin to form around them, and you'll get division of the cytoplasm, which in an animal cell is called a cleavage furrow. So you'll get reformation of the nucleus. You'll get the separation between the two cells, which is called a cleavage furrow here. It's called cytokinesis, that process. It's the separation of the cytoplasm. And the DNA will start unraveling again in the new cells. So again, prophase, you got condensing of the chromosomes. You have your mitotic spindle forming. Metaphase, your chromosomes are lined up along the center of the cell. Anaphase, the chromatids separate and move towards opposite ends of the cell. And then telophase, the nucleus reforms around the chromosomes. Cleavage furrow forms, which is cytokinesis, occurs to separate the cells in two. Okay. So it's pro, meta, ana, and tele. Okay? All right, so we'll review this again next class, but I want to show you a few more slides, and actually I might give you one kind of quiz slide. So here's just a close-up picture of cytokinesis. So cytokinesis occurs during telophase. In animal cells, it's called a cleavage furrow. In plants, it's called a cell plate but both of them are division of the cytoplasm. So cytokinesis occurs during telophase. It's division of the cytoplasm. So I want to show you a video. This is a video taken with a light microscope of a cell dividing, and it's pretty awesome that they could get a video of this. So this is kind of early prophase. You can see the chromosomes are condensing, and they're going to spend a lot of time interacting with that mitotic spindle to pull them into metaphase. So this is still prophase, but it's verging on metaphase, and you'll see as soon as they line up, they're going to separate. So chromosomes are getting closer and closer to the center. They're going to be lined up soon. As soon as they line up, okay. All right, there we go. So now we're in anaphase, and telophase occurs pretty rapidly, and there's cytokinesis, and now you have two cells. All right, pretty wild, huh? So that's cell division. You can actually see this happening. Chromosomes are so thick at that point that you can, if you stain for them, you can see them pretty well through a light microscope, and you can see cell division. So one thing you'll do if you do take 1011, part of the lab is actually looking at onion cells in the process of mitosis, and you'll be able to stain an onion cell, onion roots, and actually look at cells in these different stages. So something you might see, granted this is an animal cell. What stage of mitosis do you think this cell might be in? I think telophase, yeah. So this is very late, actually. You can see that there's already a cleavage furrow between the two. The chromosomes have reached the ends, and there's already getting separation between the two cells. How about this one? What might you call this? Yeah, I would call this metaphase. So the dark part there is the chromosomes. It's all lined up right in the center of the cell. Good. You're doing pretty well so far. Okay, here's another one, the one in the center there, very center. Yeah, so that's just prophase. If you look at the one in the center column on the bottom, that would be interphase. You can see you don't see lines going through it. It's just it's a pretty gray dot. You have a concentration of chromatin in one spot, but that's just something called a nucleoid. So the middle one would be prophase. Let's see. Okay, we have this one too. So I am going to show you all of them. But how about this one? Yeah. So I would call that anaphase. It's actually a fairly late anaphase. You can see there's a little line in the center. It's actually starting to form. That's the new cell wall. So this is really, really late anaphase, but it's anaphase. You can see they're starting to pull apart. Good. So you think you can identify those different stages? It's not all that complex. This is a little bit easier than describing to me the light reactions of photosynthesis, right? So it won't be all that complicated, right? Okay, so I'm going to stop here. That's mitosis. We'll review mitosis again next class, and then we're going to go on to another form of cell division called meiosis, and we'll learn a little bit more about that there. But definitely review photosynthesis, review all those parts that we went over today. Maybe watch these videos again.